This Week in Virology, the podcast about viruses, the kind that make you sick. From MicrobeTV, this is TWiV, This Week in Virology, a special episode recorded on October 16th, 2025. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Today I'm in Blacksburg, Virginia. And here is Virginia Tech, also known as Virginia Polytechnic Institute and State University. I'm here for a symposium, the CeZAP Symposium, Center for Emerging Zoonotic and Arthropod-Borne Pathogens. It's a center at the university, as you hear. And I gave a talk this morning about science communication, and I thought I would sit down with two virologists to give you a sense of what's going on here. And my first guest is someone who is not new to TWiV. He was last on TWiV 285. XJ Meng, welcome back.
Great to see you, Vincent.
You look the same.
You look the same too.
I know, I don't look the same.
Can't believe it's 11 years.
We sat, actually, we were in a conference room last time.
Correct.
With McDonald, right?
Sarah McDonald, yeah. She is now at Wake Forest University.
And we talked about your research, so I thought I would catch up with you. But first, what is CeZAP?
Well, as you said, CeZAP is an interdisciplinary center within the university. It's stands for Center for Emerging Zoonotic and Arthropod-Borne Pathogens. So we have 140 faculties working on all different kind of microbes. They could range from some of the social aspect of infectious disease to in-depth molecular biology of viruses, from computational modeling of microbes to public health. So it's a very, very diverse centers.
So here there is a university, is there a medical school also, and a veterinary school, right?
Correct. We do have a medical school, which is in Roanoke, it's about 45 minutes from here. And the vet school right here is on campus. And it's called Virginia Maryland College of Veterinary Medicine. So it's two state school system, headquarters here in Blacksburg, but we also have a department in the state of Maryland. So
the faculty in CeZAP, do they come from both the university and the veterinary school?
Yes, and actually the faculty in the CeZAP come from 6 different colleges. I include the Virginia Maryland College of Veterinary Medicine, the School of Medicine, College of Engineering, College of Science, College of Agriculture, and College of Environment and Natural Resources, and the College of Engineering, seven schools. It's a very diverse expertise of a group of faculty.
So you put this center together originally, right?
I did. As at the very beginning of the pandemic, early 2000, and I just felt the need to kind of get a group of people together with very different expertise, and the goals for them to collaborate.
Right.
So we decided to form this center, and the university poured in quite a bit of resources for us to start the center. And you can see here now, the center is doing fantastic. We have a very fantastic graduate program. It's called the Infectious Disease IJEP program. It's an interdisciplinary graduate education program. And so they focus on infectious disease, and it's 50 years now. We have symposium, and we have a weekly seminar series, and we have seed grant program. But importantly, the faculty now begin to collaborate. So that's the goal.
Right. It's the main goal of the center. And then
Correct.
The faculty and the students and the postdocs will collaborate, and that's always better.
Yeah.
Right? This was your idea, right?
It was a group of people at the time, and with a lot of support from quite a few folks. And I did the work, and I put a charter together and get it approved. But it was a group of faculty will get together and decide to start this.
And you were the first director also, right?
Yeah, I was the founding director for a couple of years, and but Kylen Kenhal is taking over since I stepped down, and she's doing a fantastic job. Yeah, and the center is really blossoming right now.
So Kylene will be my guest next. We'll talk to her about
Yeah.
her research as well. So I thought we could talk a little bit about hepatitis E virus. Is that okay?
Sure. Yeah,
You work on many viruses.
I do.
Did you also work on SARS-CoV-2?
I did. I did a little bit of work on that. You know, I guess a lot of virologists kind of a pivot and during the pandemic
Yeah.
working on SARS-CoV-2. But my focus, as you said, is on the hepatitis E virus now.
Okay. So what hepatitis E virus? What kind of virus is it?
This is a single-stranded RNA virus, and it's relatively small. It's about 7.2 kb in size for the genome. It's a non-enveloped spherical virus particle. It's about 30 to 35 nanometers in size, and so it's not very important but understudied virus. And each year, if you look at a disease burden, each year the virus causes about 20 million infections worldwide.
Worldwide, okay.
And leading to about 3.3 million cases of hepatitis and about 44,000 deaths. So it's a very important public health disease in many developing countries, but we do also have sporadic cases here in the United States and many other industrialized countries.
So what is encoded in the genome? I guess there's a capsid protein.
Yes, it's a very simple genome, and so ORF1 could for some of the non-structured proteins, and it has multiple functional domain, methyltransferase, helicase, RNA-dependent RNA polymers, and then there's ORF2, which code for the capsid, which is the only structured protein on the virions.
Just one.
There's one. And then there's a small ORF3 that partially overlapped ORF2 that are responsible for virus replications.
So there's a T = 1 particle?
Yeah, it is.
So it's very small, 60 copies.
60, yeah.
And the how are the proteins produced? It's not one polyprotein like polio, right?
Yeah, that's no. And the ORF2 is translated through the subgenomic RNA. And now the ORF1 translation is still kind of a debatable, and there are one school of thought is this just a one polypeptide for the non-structured protein, and they just have multiple functional domain. Now there are a couple of studies reporting that they are pretty late cleavage, but there are others that could not confirm that. So right now it's still highly debatable whether or not ORF1 is indeed cleaved, but there are quite a few studies do show there are functional domain, particularly methyltransferase, helicase, or RDRP. They are all functional.
Is there a protease encoded?
There is a protease. Yes, there is one. Yeah, and cysteine protease. Yeah.
So when the RNA gets into the cytoplasm, then ORF1 is translated?
That's correct. Yeah, for RNA virus, it's replicated in the cytoplasm. However, there are recently reports of a naturally occurring recombinant virus. And so there are human sequences inserted into the hypervariable region in the viral genome, in the HEV genome. Now, that particular recombinant virus, naturally occurring recombinant virus, do have a nuclear stage. And we did some study. We found that actually there's an insertion of human ribosome protein S17 in the hypervariable region of HEV genome. The S17 contains a nuclear localization signal. So that kind of bestow a novel nuclear trafficking capability of the ORF1 protein of the virus.
It's a gain of function.
It is gain of function,
Natural
and
gain of
naturally
function.
occurring gain of function. Sue Emerson's group in the NIH, actually, she showed that the recombinant virus with S17 insertion has expanded the host range, can infect cells from more than a dozen other animal species, not just human anymore.
Interesting.
Yeah.
So the original hepatitis E virus is just a human virus, right?
Correct. Prior to, I would say, 1997, hepatitis E was thought to be an exclusively human disease. Then our group, of course, in 1997, we discovered the first animal hepatitis E virus from pigs. So we named this swine hepatitis E virus.
It's a different virus, though, right?
It is, well, at the beginning we thought it's a different one. That's the reason why we named the swine hepatitis C virus. But retrospectively, there's not a whole lot of differences actually. They share very similar genomic organization. They share high significant sequence homology. The pig virus can infect humans. As of right now, the virus has more than a dozen other animal hosts.
Wow.
Some of them, like the virus from pig, from deer, from rabbit, and more recently from rat, has been shown to infect humans. So this is no longer just a human virus anymore. So this is a group of zoonotic viruses. And in fact, actually, the recent study that was published in PNAS, I can't remember who did it, but in that study, they analyzed more than 800 viruses, and they found the hepatitis E virus is ranked number 6 in terms of risk for spillover and zoonotic infection into humans.
So what came first, the animal Hep E or the humans?
Good questions. And if I have to guess, I don't know the answer. If I had to guess, I would say animal first.
Sure.
If you look at the diversity and all this different strain of animal Hep E virus, it has to come from an animal first, but nobody has done any study to prove
and so
that.
then it spills back from humans into animals?
It does, actually. Some of the human viruses, particularly genotype 3 and genotype 4, they infect pigs, they infect animals. But small, the other way around. And the animal virus now infect human, and that raised a lot of concern, particularly food safety concern, because we do consume some of those animal meat. That really raised a food safety concern. And in fact, actually, a recent joint FAO-WHO expert panel ranked hepatitis C as the top 3 foodborne pathogen, along with norovirus and hepatitis A virus. So it's an important foodborne virus now.
So, and wild animals are also infected, not just farm animals.
Yes, and wild boar has been confirmed to be infected, and there are virus has been identified for deer, rabbit, a number of wild animals.
Can you acquire infection by eating wild boar or deer?
There has been actually reported cases and human infection.
Because when you're processing the meat, it's not cooked yet.
Correct.
You can infect yourself.
And actually in Japan, there are quite a few reports. There's one report, remembers, in Lancet, and showing that consumption of wild boar meat and 4 people infected. And some of the pork products also. And one of the very nice study was done by the group from France, and they showed that the fugitello, there's a pork sausage, and this smoked sausage, so it's undercooked, and people who consume those fugitello sausage develop hepatitis.
I talked to a guy in Zurich many years ago who likes to hunt wild boar, and he works on Hep E. And I said, well, how do you know? He likes to eat it. He says he just does PCR before he processes it to see if it's infected.
Yeah, I mean, there, a lot of folks, they eat those raw, and they don't cook it all. And many parts of the world, they consider this that as delicacy. So
do most human cases? How do most human cases arise? Is it human-to-human transmission, or
very limited human-to-human transmission, and the majority of the cases are through contaminated water, contaminated food, which has become an increasingly a major problem, especially in Europe. And foodborne hepatitis C in the last 5-6 years has been really becoming a problem. For example, in Germany, each year, thousands of cases of a foodborne hepatitis C in Germany. Contaminated water and contaminated food.
So when you ingest the virus, where does it go? From your stomach or intestine?
The virus, of course, go to the stomach and then go to the GI tract. So the virus initially replicate in a small intestine.
So it's very resistant to stomach acid.
Right, it does. And then replicate in the GI tract, and then goes to the bloodstream, reach the target organ, which is the liver. The replication in the hepatocytes, the levels are very low actually, and there are quite a few studies show the virus actually replicating more in the bile duct epithelial cell than in the hepatocyte, and they also replicate quite efficiently in the epithelial cells of the small intestine.
But the And so those are the main cells, the hepatocytes, the bile, and the small intestine. No other sites of replication that we
There
know?
are some other side, and there has been, we have done a study, and we actually, I think we found some other tissues that are susceptible to the virus infection, and I think kidney is one of them, and besides the liver, small intestines, and which kind of explain now, the virus, in addition to cause hepatitis, it is also now linked to a number of extrahepatic manifestation disease, including some of the neurological sequelae, actually. The HIV has been linked to quite a number of neurological complications, Guillain-Barré syndrome, neurologic amyotrophies, and so both the central nervous system and the peripheral nervous system apparently are affected by the virus.
Do you think the virus is getting into the nerves?
There are studies, we published a study recently which show actually the virus can cross the blood-brain barrier, and we also have evidence we haven't published yet that the virus replicate in some of the cells in the neurovascular unit.
So you can probably do, organoids, neural organoids, and that sort of thing.
Actually, we are collaborating with Colin Bishop and Michael Seid, and Tony Atala at Alderman Wake Forest University. We're trying to using some of the organoids they produce and to test some of the susceptibility and also determine the infection dynamic of the hepatitis C virus.
But the main symptom is hepatitis, right?
Correct. And most people have acute viral hepatitis, and there are some individual do progress into chronicities, and those are usually immune compromised individuals, particularly organ transplant recipient, patient with HIV/AIDS, patient with lymphoma or leukemia. They do develop into chronic hepatitis. In fact, some of those organ transplant patients, up to 80% of them, if they infected, they develop chronic infection.
And this is lifetime?
It is not a lifetime, and they do after, I don't know how long. I don't even know if anybody look at how long they will persist for the chronic infection, but they do develop chronic infection, and fortunately we don't have an HIV specific drug, and so the clinicians in the hospital, they are now using pegylated interferon alpha and also ribavirin and off-label to treat the hepatitis E.
And so do these patients ever develop cirrhosis and hepatocellular carcinoma?
Some of them they do, particularly cirrhosis, not very common for HCC, like hepatitis B or hep C. They are more common, but hep E, we do see cirrhosis, but not in HCC.
Is the blood supply checked for hep E?
It has been checked, and there are reports, like in Japan, they found the virus, but a very small percentage of the blood donors are positive for hep E. In the U.S., there are, I think, at least 2 studies. They did not find any positive donors, and so, which is not surprising because the viremia in those infected patients are very transient, lasts probably only a couple of weeks, and so blood transfusion, contamination, probably not on a major route of transmission, but it does occur. It has been reported in other countries.
So probably when you're in the acute phase, you don't feel good, you're not going to give blood.
Correct.
Right. So the blood in the U.S. is not checked for hep E routinely?
No, and largely because we don't have an FDA-approved diagnostic test, unfortunately. And so all the tests we have so far are, homemade diagnostic assays.
So no company is interested in doing this?
There are some tests in overseas company, and they're just not, for whatever reason, FDA licensed test.
So would this be a concern in a solid organ transplant patient?
Definitely.
Would you test them for hep B before?
I would. If if I'm in a surgery, if I'm in a transplant surgeon, I would do that, because if they're infected, 80% of those organ transplant patients, if they're infected by the hep E virus, they're gonna develop chronic infection, which is a major problem.
So does this, once you're infected and recover, do you get it again?
Good question. I would say there's going to be pretty good protection if you get infected. There's only one serotype so far, and one serotype hepatitis C, you would assume that's going to be cross protection, and even if you're going to infect it with a different genotype. But I don't recall I see any study look into that, and reinfection, whether or not they can be protected though. But the vaccine, there is one vaccine that's licensed now for use in 2 countries, and that vaccine so far has shown very high efficacy. And they published in the paper in Lancet, they show more than, I can't remember, 95, 96% efficacy. So that's a pretty good vaccine. But unfortunately, it's only 2 countries has licensed.
What kind of vaccine is it?
It's the recombinant vaccines. I think it's E. coli, just recombinant capsid protein based.
In E. coli?
Yep.
So they eat it?
I think the injection.
Oh, so it's a purified capsid protein. Okay.
Yeah.
Because there's some bacteria you can eat.
That's correct. Yeah.
Spirulina, for example, you can express proteins, antigens in them.
Correct.
And just eat them. That would be interesting because the target would be the gut, I would guess.
Yeah.
That's interesting. And the only therapeutics are ribavirin and interferon.
Yeah, off-label use, so it's not HIV specific. Yeah, and all the side effect of ribavirin and some of the things. So we do need an HIV specific antiviral development.
Do you think we also need a vaccine in the U.S.?
It would be nice, but
maybe it would be a travel vaccine.
Travel vaccine, or some of those high-risk patients, organ transplant recipient.
Yes.
For the general population, probably not here in the U.S., but in some of the developing countries where this disease is prevalent, I would hope we're going to have a vaccine and to prevent those diseases there, particularly in those developing countries, because waterborne and contaminated food.
Sure. Are the number of cases increasing every year?
The foodborne is increasing. I don't know if it's because we now have a better diagnosis and better awareness, or this real increase. I think it's probably because we know more, we'll tend to look for more, and that's the reason why maybe we see more cases. It doesn't necessarily mean the cases are increasing. But there's thousands of cases each year in some of the countries. That's a lot.
You said there were how many total globally?
20 million infection per year.
That's a lot.
That's a lot. That's from the WHO figure.
And deaths are 40,000?
44,000 deaths each year. And so it's an important public health disease.
And I guess it varies from country to country. So
correct.
Not every country would need a vaccine, right?
Yeah, the heaviest disease burden is in those developing countries.
So now this recombinant, this naturally occurring recombinant, it has a ribosomal protein gene inserted, right?
Right.
Is this starting to take over, or is it just limited?
No, it's very small percentage in the quasi-species population, very small percentage in that patient. It's from an HIV/AIDS patient, actually. It was the work that was done by Bob Purcell's and Sue Emerson. They were able to isolate this virus, and they sequence them. They say there's a natural ribosomal protein S17 insertion there. Now, there are other groups now showing that all kind of other human sequences inserted in the hypervariable region. S19, there are some others sequences also inserted in that hypervariable region. It's a very unique region. We did quite a few studies looking into this hypervariable region. It's rich in protein residue, but the sequence is just so different. Between 2 genotypes, they differ by as much as 70% sequence.
Where in the genome? Is this ORF1 or?
It's the ORF1, yeah.
So the HIV patient probably had a chronic infection.
Yes,
so.
It does, a persistent infection, yeah.
So many years ago, Bert Semler described a recombinant poliovirus from cells in culture where it had picked up some ribosomal sequence. And he theorized that the polymerase and the ribosomes collided on the same RNA, and that's how you got, so the polymerase copied some of the ribosome and then went back to the genome. And so that's how, maybe it's a similar thing.
It could be,
right?
Could be.
It's really interesting.
Yeah.
But there's no evidence that it's any more virulent than
no, I mean, it doesn't appear that's the case. Yeah, and also it's not just one unique specific human sequence. There are I think 4 or 5 different human sequences now has been found to be inserted into the hypervariable region. Yeah.
Is there an animal model you can use to study this?
We do. There are quite a few. One of them is the pig model, and the pig model are highly susceptible to genotype 3, genotype 4, both of them infect human. There is the chicken model, but the chicken model is only susceptible to the chicken hepatitis C virus.
Okay.
We now have the rabbit model, and we actually isolated the rabbit strain of hepatitis C virus from the rabbit tree, actually right there in Virginia here.
Mm-hmm.
And we were able to actually establish the rabbit model for this virus. So there are quite a few models now.
But not mice.
Not mice, but gerbil. And so
Okay.
a different kind of rodent. And gerbil has been shown repeatedly are susceptible to 3 at least different genotypes of hepatitis C virus. So that could be a very valuable model system. Now, the problem with using gerbil, as you know, Vincent, is the gerbil develop those seizure, those naturally occurring seizure in those animals. So it's not a really good animal model system. You know, there once they develop seizure, they affect some of the experimental.
But they're also outbred, right?
Right.
Do you know the receptor for the virus?
No, and people has been looking for this, I think for probably a good 20 years, and no one has found the receptor. So that's probably one of the bottleneck for this field, and we don't know the receptor for hepatitis C virus.
Because if you had, then maybe you could make a mouse model, transgenic, right?
Like you did
Yes.
with the PVR, yeah, definitely,
Yes. Long time ago.
Yeah.
That would be, but the other animals are hard to work with, pigs and chickens, even rabbits. They're not inbred either. Right?
No, it's difficult, especially some of the large animal, and you can't use large numbers. So there are. Definitely.
So for pigs and chickens, are there veterinary vaccines or no?
They don't. See, the chicken actually is associated with a disease. It's called hepatitis splenomegaly syndrome, and so it's caused actually cause a lesion in the hepatitis, enlarge the liver, and the hemorrhage in the spleen, and a portion of infected chickens. And there's egg drop, and so if you're a poultry producer, you don't want to have an egg drop syndrome from your chicken farm.
Right.
But in pig, it does not cause overt clinical disease at all in those infected pigs. And even though they do induce microscopic lesion of hepatitis, you see under microscope, the infected pig do develop hepatitis, and you see the foci necrotic hepatitis, you see the lymphoplasmacytic hepatitis. So they do develop lesion, but not disease.
So you don't know if your pigs are infected.
No,
basically, you'd have to screen them.
You have to screen them, and unfortunately, a large number of pigs in the United States and many other countries are infected by the virus. So that's really caused a problem, a huge concern for food safety.
Yeah, of course. Because
Yeah,
you could acquire it from pork.
yeah. We did a study a few years ago. We in collaboration with the USDA, we found about 6% of the pig at the slaughterhouse. That's the time they're mature, and they're still viremic. You can imagine that 6%, the blood is going to contaminate the food supply chain.
So the people who work in slaughterhouses, are they antibody positive?
Nobody tests them.
I bet
there's some
right?
of them will be. And we did test it. A group of pig veterinarians, and there's a study we did probably 20 years ago, and we showed that that veterinarian who work with the pigs has significantly higher seropositivity compared to the controls.
They may also have hepatitis, right?
Yeah, and it's probably subclinical infection, and they just don't know they have an infection.
You have a lot of pigs in your office.
I do.
Lots.
You can see I love pigs.
You work on other viruses that infect pigs too, right? All those
yes, we do. And sometimes I guess people knows, and you think some of the folks, the students, suppose that they go on a conference somewhere, they found a pig, they buy it from me.
That's funny. Yeah. So this rat, you wrote a review article on rat hepatitis E virus. Would you say it's really different from human hepatitis E virus, right?
Yeah, the rat, that review article was actually done by, I think the first author, former postdoc, Kush Yadav. He just started his own lab actually now at Ohio State University. And he worked with Scott Kenny, who is an associate professor at Ohio State. And so I was just one of the co-authors there. Scott is also a former lab member here. He did a postdoc with me. So yeah, Kush wrote that paper, and so that is relatively new, and it's the rat virus. It's called C1, HEV-C1. It has been identified in rat population. It has been shown to infect human. There are, I can't remember, I think at least 20 some cases of human infection by the rat HEV has been documented. I know there are 2 cases in Canada. There are some cases in Hong Kong. So potentially it could be an issue. And maybe we haven't actively looking for those cases.
But its sequence is diverged from Hep E.
Very different. Only about, I would say, about 50% sequence homology was the human HEV. So they are very different, and yet they can infect human.
Yeah.
So
that's very interesting.
Very interesting. Yeah.
And maybe the receptor is different. Maybe it's the same. Would be interesting to know all that.
Yeah, definitely. Right? And there's a, if you look at all those animal Hep E viruses, really, their biology, their natural history, and their pathogenesis is grossly unknown. Yeah, so there's a lot of work that need to be done. I wish more people are going to work in this field. This hepatitis E field is still very small.
Yeah, you said it's understudied.
Understudied, extremely understudied.
How many labs do you think there are?
In the United States, I probably can count it with my two hands. Yeah. So fortunately, there are some big names in the field that begin to, devote time to study this virus now.
What other things do you work on with Hep B in the lab right now?
So we have a multiple project, and one of them we look at one of the very unique features associated with the hepatitis virus infection. That is a high mortality and severe disease during pregnancy, and the mortality rate can be up to 25% in infected pregnant women.
The mortality of the woman or the fetus?
Both, actually. Both? There are neonatal mortality, fetal loss, and the mother develop formal hepatic failure. So we're trying to look at underlying mechanism leading to the development of those severe disease during pregnancy. So we'll focus on both the viral genetic element and the potential host factor that may contribute to the severe disease. And so that's kind of one aspect, one area we're working on. The other one is we'll look at one of the extrahepatic manifestation disease, the neurological sequelae. As mentioned earlier, there are quite a number of neurological complications, and we'll try to find out why. So we're using some fancy tools. Now we have a brain organelles. As I mentioned, we collaborate with Colin Bishop, Michael Seed, and Tony Atala from Wake Forest University, what they call Institute for Regenerative Medicine. So they produce this beautiful brain organelles, and we're one of the postdoc, Hannah Brown, in the lab is studying that aspect. We'll try to find out what are the underlying mechanism leading to the infection in the brain, so that's another direction we're working on. And the third direction we've been working on is trying to understand why some animal strain of hepatitis E virus jump species infecting humans, but others don't. So for that direction, we focus mostly on some of the viral genetic element that we think may be responsible for cross-species infection. And I think we've been down this quite a few years. We kind of narrowed down to in a particular regions in open reading 1, that's the hypovariable region. We have evidence that this particular region, we think, contributing to the host adaptation and cross-species infections.
And this ORF1 is
non-structural proteins. Yeah. It's kind of odd because the beginning we never thought it was ORF1. We thought it's the capsid.
Yeah, receptor interaction.
Exactly. You would have thought the capsid bind to the receptor and determine host ring. And we didn't study. It wasn't the case.
So is most of your effort on Hep B, or are there other viruses that have more?
Yeah, well, mostly right now, I think the last 5 years, I kind of made a conscious decision and to focus on my favorite virus, that's the hepatitis C virus.
Okay.
And we have a cover project on a Porcine epidemic diarrhea virus, and pig coronavirus. So collaborate with Steve Zogner from the University of Virginia Medical School. We have a project that is funded to develop vaccines against the porcine coronavirus. That virus caused a devastating disease in piglets and very severe diarrhea and dehydration.
Right.
And, the dehydration, #1 cause of death in those young animals. So we'll try to develop a better vaccine. Steve has this beautiful, actually, vaccine platform. And he has this, I think he called it synthetic biology vector system. So he engineered the bacteria, and he was able to delete about 30% of the surface gene of the E. coli, and yet the bacteria is still viable. And then he cloned some of the foreign gene in the surface. So he had this beautiful system. So he applied the grant, and I was the co-PI on the grant. We'll get funded. So work on that.
Okay.
Yeah, and we have also collaborate with the engineering actually on campus, Mike John. He's a biological engineering. It's a very sharp guy, and so he used this nanoparticle delivery system. So we have 2 grant funded to work on the PEDV vaccines using this nanoparticle delivery system. He can engineer the vaccine and just make it just look like a viral particle of VLP.
Cool.
And so that kind of a side project for that virus. I'm not leading it. It is some of the collaborators they are leading.
So last thing I want to ask you is, science is in a tough time in the U.S. now. NIH is a mess, and its funding has been interrupted. Indirect costs are cut, and CDC is being destroyed. How do you view this, the future? Does it look bad, or what do you tell young people who want to come in the field?
Yeah, it is a tough time right now, and what we're facing here today in the States is tough, and especially for some of the junior faculties and grad students and postdocs. But I would say, at the end, science is always going to prevail, and I would not give up. And some of the junior faculty, they tell them, try to maybe diverse your funding sources. There are other sources still funding science, industry, nonprofit, some of the foundations. So you can still do your work. And we just recently signed a contract with the pharmaceutical company. And so just don't give up. And other than that, I don't know what else to do.
You wouldn't leave the U.S., would you?
I would not.
Because your former country, China, is amazing in research now.
They're doing pretty well.
They recognize the value of investing in research. Independent of politics, right?
Yeah. And I built a career here at spend more time actually my life here in the States than in China. I came in here in a very young grad students, and it is just a very challenging time.
I think people who want to be scientists need to pursue it, right? They can't be discouraged.
No, they have to. It's kind of like we're doing research. We're going to have probably tons of failure, and by the end, you prevail, and so it was kind of funny. I was chatting with my daughter, and she's a second-year graduate student, and some of her experiment didn't go the way she wanted, and so that's part of research.
Of course.
If you work for the first time, there's no such thing as a research. So
that's why it's called research.
Exactly.
Right? Do it over and over and over again.
Yeah.
Well, that's part 1 of TWiV from Virginia State. My guest, XJ Meng. Thank you for joining me today. I appreciate it.
Well, thank you. I appreciate that you come here. This is your 3rd time, I believe.
Yeah, I like it here.
Yeah.
It's nice.
Maybe we should get an office for you here at the Virginia Tech.
So stay tuned for part 2, which will be coming up right now. See ya. Hello, everyone. I'm back for part 2 of TWiV from Virginia Tech. And now my guest is Kylene Kehn-Hall. Is that the way you say it? Kehn?
Ken.
Kylene Kenhal.
Yes.
Oh, it'd be like K-E-N. Professor and Director of CeZAP. Welcome to TWiV.
Thank you. Great to be here.
Did you, so you came here to be director of CeZAP originally, right?
No, I got to inherit that once I came.
Once you were here.
Yeah.
So let's go way back to the beginning.
Mm-hmm.
Where are you from originally?
Originally? Where was I born originally? I was born in California. My father is a retired Marine, so we moved around a bit when I was younger.
Yeah.
And then 6th grade, we settled in Fredericksburg, Virginia. And I'd been there most of my life until I came to Virginia Tech.
So you're Virginian, really?
I am a Virginia girl. Yeah.
Now, where did you go to college?
I did my undergrad at Virginia Commonwealth University in Richmond, Virginia.
Mm-hmm. VCU, right?
VCU, yeah.
And did you major in science already?
Biology and chemistry. So I have 2 degrees, actually, one in biology and one in chemistry.
Why did you become a science major? What made you do that?
Yes, so when I entered undergrad, I was undecided at first. I really had no clue what I wanted to do, and it was just a general biology course that caught my attention. And I thought, you know what, I really love this course. Let's go with it. It was really not much thought into it, and I became a biology major and didn't have a lot of guidance in undergrad about what does that mean when you graduate, right? And so when I graduated, I really couldn't find a job, which I think a lot of undergrad biology majors fall into unless they're going to medical school or they've done research while they're in undergrad.
So what did you do when you decided to go right to grad school?
I'm going to go to grad school, yeah. So actually at the time, I really wanted to do forensic science. And I applied to a couple forensic science programs, didn't get in.
Is it because you watched NCIS?
It was big then, CSI was big.
What was her name, the forensic scientist?
Abby
Shudo.
Yeah, Abby Shudo on NCIS.
So you wanted to do what she did, right?
I really like that show. Yeah, I actually minored in criminal justice in college. Cool. And I did not get into forensic science programs, which I think was a blessing in disguise. And I ended up applying to more general programs in like biochemistry and molecular biology and got into those. And so I ended up going to George Washington University and doing both my master's and my PhD in biochemistry and molecular biology.
Did you start working on viruses back then?
I did. So I entered a retrovirology lab. So Fatah Kasanchi at GW took me in and taught me about research.
What did you do? Which retrovirus did you work on?
In my master's, I studied human T-cell leukemia virus, so HTLV, and I'm trying to understand how, cells were transformed and what, some mechanisms behind that, looking at cell cycle regulators primarily.
We just did a paper on TWiV where they made a mouse model for HTLV1, humanized mouse, and they tested some antiretrovirals to see if they could be useful because apparently they're not really well studied for HTLV, and a lot of people are infected and they could use it. Right? And then for your PhD, did you work on some other virus?
So I did some side projects on HIV when I was in the lab, but my dissertation was actually on breast cancer. So I was in a retrovirology lab doing breast cancer, but I was continuing looking at cell cycle regulators. I had a DoD predoctoral award to focus on breast cancer.
Okay, so then you went back to virology as a postdoc, I presume?
So I've kind of had a weird career. I've done different things. I actually decided to go to the FBI and work in their forensic science and counterterrorism research lab.
Should I be worried that you're ex-FBI?
I did not officially work for the FBI. I was a contractor. I had an ORISE fellowship, and I thought, let me try out forensic science. I see if I like it. Turns out
I
did not like it.
Okay. How long were you there?
About 1.5 years.
Okay.
And then I basically got pulled back into virology. I went up to USAMRIID, U.S. Army Medical Research Institute of Infectious Diseases, and I worked in Cenovirus lab up there, and that's where I fell in love with the really dangerous viruses.
Now, USAMRIID is in Frederick?
Frederick, Maryland.
So I think we visited. I think we did a TWiV and a TWIM there.
Yes, I've seen that one.
And we were going to do one at Walter Reed, but now that the government shut down, we have to wait for it to be turned on again. Oh, that's interesting. So then, before you say, I was going to ask you something about, I forgot, it left. Anyway, so you did a postdoc on the dangerous viruses. Yeah, because they have a BSL-4
They
there.
have BSL-3
Actually, there are
and
two,
BSL-4.
right? There's one in US Amrit. Is there one at
UTMB also is a BSL-4.
no in at the same near US Amrit? Aren't there two?
Oh, and back.
And
yes.
back, right.
Mm-hmm.
I wonder if those are shut down for the government shutdown, right?
I'm not sure.
Yeah. I knew a guy who worked there, Jens Kuhn.
Mm-hmm.
You know that name?
I do know the name. Mm-hmm.
And he was on TWiV a couple of times, and then he left, part of the government purge, unfortunately.
Mm-hmm.
So what did you do after that?
So I did a short transition back at George Washington University. I received a research assistant professor position there. And then I had the opportunity to start my own research lab at George Mason University. So I started there as assistant professor in 2009.
What were you working on?
So there I got to develop my program in emerging RNA viruses. And at the time, they have a NIH-funded biomedical research laboratory there. And it hadn't quite opened yet. So when I started my position, they were actually the building was under construction still. And so I got to work very closely with safety and develop their select agent program there. And I decided the first virus I wanted to work on was Rift Valley fever virus.
Which is your favorite, right?
It is my favorite.
But you work on other viruses.
We do work
now.
on other viruses. Yeah. So we work on Venezuelan equine encephalitis virus and Eastern equine encephalitis virus.
The encephalitic alpha viruses.
Yes.
Right.
Yeah.
And then you came here after that?
So I came to Virginia Tech in 2020, right in the middle of the pandemic.
Middle of the pandemic?
Mm-hmm.
It must have been tough, right?
It was a little difficult, but I think it was a great move for me, but I was also able to keep my lab going a little bit at George Mason. So I had some students there that were finishing up their work so we could still do some work in containment. And then while I got my program up here running. So it worked out well. So
you came here and then you became director of CEZAP.
So when I first came here, XJ Meng asked me to be on the internal advisory committee for CEZAP. And I did that. And then about a year later, he asked me to become the associate director. And I served in that role until 2022, when I became the director of CEZAP.
Okay. And you're right. This is XJ's office, but you're right next door,
yes,
right?
Exactly. Yeah.
So tell me in your words, what is CEZAP?
Okay. Well, CEZAP stands for Center for Emerging Zoonotic Arthropod-Borne Pathogens, so CEZAP. We were trying to come up with like a catchy title.
So you threw arthropod in there because you didn't have to, right?
We didn't. Well, we had, I think initially we were thinking vector-borne, but it didn't, the acronym didn't quite work. And so the A worked better. For the CEZAP, which I think is funny for
you
mosquitoes.
See you guys sitting around a table and brainstorming.
Oh, there was a number of us trying to figure this out. Exactly. And so what is CEZAP? CEZAP is it's a virtual center, but we have over 130 faculty members across 7 different colleges at Virginia Tech. And the idea was that we want to pull together any researcher who's interested in infectious diseases and help provide them with resources and different opportunities to develop multidisciplinary research. And we wanted to make sure that we're pulling together like engineers, for example, and how can you work with your classical virologists, pull together computational biologists? How do you work with bacteriologists? And anyone that wants to be part of it can be, and we hopefully facilitate great collaborations.
Yeah, that's the key. It's all about getting people to work together who would not normally reach out to each other.
Exactly.
And facilitate it. And then the students get involved with postdocs, and it's all, and as you saw this morning, we were at the symposium, and a lot of people in that room.
Yeah.
A lot of enthusiastic people.
Yeah, it was great to see. Yeah.
And in the meantime, you continue your research, right? You have your lab here. So I wanted to talk about your favorite virus a bit.
Okay.
Rift Valley fever virus. So what is Rift Valley fever virus?
So yeah, it is a mosquito-transmitted virus. I will probably say RIFT for short. So it's transmitted through mosquitoes to humans and as well as to livestock. And it causes very severe disease in livestock. This can look like a hemorrhagic fever. It's classified as a hemorrhagic fever virus, so it can cause a lot of liver disease. But what classically happens is when animals become infected, you see these abortion storms that come through and tend to that's like the first warning sign that you may have a rift epidemic happening.
So livestock would be cattle?
Cattle, sheep, goats, primarily.
But not pigs?
No, not pigs.
And where do the mosquitoes get the virus from? Another cattle, or is it from other reservoirs?
They can pick up it from cattle or other livestock, but they also maintain it within themselves. So the female mosquitoes will pass it to their eggs in a process called transviral transmission. And so this keeps it in the mosquito population.
That's interesting. And the mosquitoes are okay?
Yes, as far as we can tell.
Of course, you can't ask them how they feel, right? They may feel badly.
Yeah. I mean, let's say in cell culture, it doesn't really cause cell death of mosquito cells versus a human or another mammalian cell. It causes extensive cell death.
That's interesting. I never heard that it maintains in mosquitoes like that. Usually there's some other reservoir.
There's definitely, people have looked for other reservoirs. You can find it in some wildlife as well.
So do most cases occur in areas where the livestock are infected?
Yes, for sure. So you see that. So it's found in, I should have said this, in sub-Saharan Africa primarily. And so it can really devastate the livestock.
What's the mosquito, Culex species?
Both Aedes and Culex species.
So is it ever in the U.S.?
We have never found Rift in the U.S., but we do have mosquitoes that are capable of transmitting. So there's been a number of laboratory studies that say, like, the mosquitoes that we have, can it actually transmit? And they can.
So it could be imported and established like other things like H5N1 in cows, right?
Yes.
It could happen.
That is the fear, that will happen. The other fear is that it would establish itself perhaps in our wildlife population. So there was a study done by Jürgen Rick's group where they looked at, the susceptibility of white-tailed deer, for example, to Rift. And they found that they were highly susceptible and it caused death.
You see, there was just a locally acquired case of chikungunya in Long Island.
I did hear that.
So that's interesting because she didn't travel, and so where did it come from? Maybe there's some virus somewhere, right?
Some low level circulating. Yeah, we don't know.
You always have to worry. So when people get infected, what is there a long incubation period?
I think it's around 5 to 7 days for humans, maybe 3 to 4 in terms of incubation. And the human disease is luckily milder than what we see in livestock. But it is a spectrum of disease too. So you can get the liver complications. It's a bit biphasic. So initially you get more of the peripheral disease, and sometimes people even feel better. And then they develop encephalitis because the virus has made its way to the brain. You can also have ocular complications. So sometimes you have blindness that happens.
So it's conceivable that someone could get infected in Africa and come to another country while still incubating, and then but then they would have to. I presume there's a viremic phase. That's where the mosquitoes get virus, and so they'd have to encounter a mosquito. It's a lot of rare events that have to happen, but you never say never.
And luckily, a lot of the human cases are subclinical, we think, right? So that people get it, or maybe they're just a little bit febrile. They're not the full blown, really severe disease.
So we're not too worried about it in the U.S.?
Oh, we're still worried about it. Yeah, we're still worried about potential introduction because of all the different reasons that can happen, right? You get mosquitoes that are accidentally imported into U.S., et cetera.
So what containment is it worked on?
It is BSL-3, so biosafety level 3, and it's also a select agent. So we have an increased biosecurity.
Your FBI comes in, right? You have to have FBI clearance.
We have to have, an FBI. It's not a clearance. I should remember the word for it. It's you have to be SRO approved, essentially. Yeah.
Because I know for SARS-1 you have to have FBI.
Yeah, I mean it goes to the FBI, but it's not your top secret clearance. It's a different terminology for it.
Okay.
Yeah.
And your work didn't already have that, so you had to go through it.
Yeah.
That's great. So there is a BSL-3 in this building?
Not in this building, but we do have actually multiple BSL-3s on Virginia Tech's campus. We have our newest one is in the Roanoke campus, and they started SARS-CoV-2 work on that campus. And the BSL-3 that I work on is been at Virginia Tech many years. I don't remember how, but it's been here for quite a long time. And we have an in vitro BSL-3, and then we have an ABSL-3. We can do animal work. So
what kind of animals are models for this virus?
A lot of people work with mice, so mice can be really good models. They're very susceptible to the virus. And the mouse model we work with is BALB/c mice, and they do develop the biphasic disease. So you can see some liver disease, and then you can see encephalitis.
So how do you inoculate the mice?
We can do either subcutaneously to kind of mimic a mosquito bite,
okay.
or if we really want to study the encephalitic form, we might do it intranasally.
And that predisposes to encephalitis.
It does, and you'll get almost like uniform encephalitis if you do intranasal.
Interesting. So you think it goes through the olfactory sinus?
Yeah, there's not a lot of studies that have shown how Rift enters the brain so far, but I think it's possible that it could go up through the olfactory bulb. Yeah.
So if you get infected, either a mouse or a human, do you get reinfected or you get immunity?
That's a good question. I would imagine you have some immunity, but I haven't followed those studies as closely. Yeah.
There's no vaccine, right?
There's no vaccine, but the vaccines that are under development, neutralizing antibodies are pretty important for those. But there's no human vaccine.
Right.
There are some veterinary vaccines that are used in Africa.
Because it would seem like in parts of Africa where there's a lot of disease, a vaccine would be good.
Yes, there's been one called Clone 13 that they've used in South Africa for many years.
And there are no antivirals that can be used either, right?
No, unfortunately. Yeah.
Okay. But you said the human infections aren't so severe, so it's just the animals.
They're not, except for when you get the severe form.
Yeah,
so if you
right.
do get the hemorrhagic fever form, you have about a 50% chance that you'll die from it.
So not only is Rift Valley fever virus, or Rift, your favorite virus, but it encodes a protein that's your favorite protein, right?
Yes.
NSS?
The NSS protein.
So that's non-structural?
It's a non-structural
And what does
protein.
the little s stand for?
It's encoded off the small segment of the virus. So the virus has 3 segments, a large, a medium, and a small.
Is there a negative stranded, right?
Negative.
So this is an enveloped virus
Correct. Yeah.
with glycoproteins in the envelope. Do we know what the receptor is for the virus?
There's been a couple that have identified. DC sign has been shown to be important. Work out of Amy Hartman's group has shown LRP1 is a entry factor. That's probably the most recently identified one.
So why is it your favorite protein? Because you work on it a lot?
No, I was really fascinated by the protein when I first started working with Rift. It has a unique aspect to it, so that the virus replicates in the cytoplasm,
Right.
but NSS actually will go into the nucleus. And once it enters the nucleus, it forms these really large filaments. And these filaments will just wreak havoc on the cell. And so I was really interested in understanding, what's the importance of these filaments? What is NSS doing in the nucleus? And, it's a cytosolic replicating virus, so what is it doing in the nucleus, right? And so the general answer to that is it's an interferon antagonist. So this protein, its role is to shut down the interferon response, but that's just one role of it, right? It does a lot of other things within the host cell.
Is it essential? Can you take out the gene and?
You can delete NSS. The virus will replicate the same in interferon deficient cells, but if you put them in interferon competent cells, you'll get reduced viral titers. In fact, some of the vaccines they delete this virus, or this viral protein in some live attenuated vaccines.
No, okay, so it will the virus replicates without it, but you need it in the context of interferon, basically.
Yeah.
Right. And so one of the things it does in the nucleus is to interfere with interferon transcription. Is that correct?
It does. Yeah. So
specifically, or does it do a general shutdown?
There's been studies to show some specific mechanisms by which it suppresses interferon transcription, but for sure it does impact other transcription. So it sequesters and also degrades some of the general transcriptional machinery. So some of the TF2H components, they're actually, NSS will link up with the proteasome and shuttle them for degradation.
Right. And polio does that as well. Polio shuts down Pol 1, 2, and 3. Is this just Pol 2 specific?
Pol 2.
And so I assume you've done RNA-seq and looked at everything that's shut off, right?
We have done RNA-seq. It was actually a difficult study because the other thing that NSS does is it blocks mRNA export as well. So it's, as I say, it's a very fascinating protein that it tries to shut down multiple arms of the immune system. And so what we found in that study was, of course, there were a large number of genes that were down, but we still had some genes that were up regulated, which we were surprised, but they weren't making it to the protein level because the mRNA was being blocked.
So what's the mechanism of that? It must be something to do with the nuclear export machinery, right?
Yes, but actually that's not well known. Yeah.
Polio does that too. It cleaves some of the NUPs.
Mm-hmm.
So maybe that's something to look at.
NSS does interact with NUP98, but that was more so thought to be important for it actually getting into the nucleus. Yeah.
So you also, in a paper that I looked at, you look at the interaction of NSS with autophagy. Can you tell us about that? What is autophagy?
Sure. So autophagy, I like to think of it as like the cellular self-eating system. So essentially when the cell becomes stressed out, it pulls in its own components in order to degrade them into individual packages. So, proteins are degraded into individual amino acids to provide resources for the cell to continue growing. And in the case of viruses, it often can be used to basically take up viruses and also degrade them. So it can be used in an antiviral manner.
So it's an early pre-interferon. We call it intrinsic, right? Because it's on all the time in cells. So does NSS interfere with autophagy, or does it stimulate it, or does it do both?
Yeah, so we found that it interferes with autophagy.
Okay.
So what we found was that NSS had this 4 amino acid stretch in it, which is called an LIR motif, or an LC3 interacting region. And so we found that this specific region enabled it to interact with LC3, which is a key kind of mediator of autophagy. LC3 helps to bring cargo into the autophagosome. And what NSS does is actually keeps LC3 in the nucleus and prevents it then from exiting the nucleus in order to kind of participate in the autophagy process.
So there are more than one LC proteins, right?
Yes, there's a family of them.
I think this protein interacts with all of them, right?
It does. Yeah, that was pretty nice. So there's like 2 subfamilies. There's LC3, ABC, and then there's the GABARAP family. So GABARAP L1 and L2. And yes, NSS was found to interact with all of these.
Your paper has a lot of acronyms in it.
I'm sure.
Besides the LCs and lots of all these other things. But what is LC3 doing in the nucleus?
LC3 tends to stay in the nucleus until autophagy is induced. And so in our system, you can induce autophagy through serum starvation, for example. So the cells get stressed, and then you'll see LC3 get exported into the cytosol. And so
the protein is actually in the nucleus?
It is, yeah.
It's weird, isn't it?
I think it's a way that the cell wants to basically regulate autophagy.
Because it has to be made first in the cytoplasm, then it goes in the nucleus. It must have an NLS, right? LC3.
Yeah, it's a very small protein, but I would imagine it doesn't, I don't know if it has an NLS.
And then it goes back out again.
Yeah, so it gets exported by a protein called DOR. Yeah,
another one. And so NSS binds to LC3 and prevents it from importing material into the phagosome, the autolysosome. What is it called? An autophagosome?
Autophagosome.
Autophagosome. Do the autophagosomes still form?
We don't see a lot of autophagosome formation with the wild type, we use a attenuated strain, but with the parental virus, we do not see that. But if we use a virus that has an amino acid substitution in that motif, that's really critical for that interaction, then we see autophagy turned on and the autophagosomes formed.
And so if you have a null NSS virus, then you don't see this antagonism of autophagy. Is that right?
We haven't done that experiment, actually. We did it with just the specific amino acid substitution to really link it to that particular motif.
So you interfere with NSS binding to LC3 just by a few amino acid changes, which is probably better, actually, than a null, because there can be so many other effects. In that virus, it does not. So autophagy proceeds.
In the amino acid substituted, correct.
And what's the effect on virus reproduction? Is it decreased?
We see about a log and a half, log 10, log and a half reduction in viral titers.
And does this happen early enough so that it would make an impact on, say, disease in an animal, do you think?
I believe so. We have some unpublished preliminary data that this particular virus causes significantly reduced disease, at least in an animal model, in a mouse model.
It could be a vaccine candidate, right?
It could. Right now, we made this amino acid substitution within the attenuated strain already, so it's like an additional added layer of potential safety on top of that.
Right. And in this paper, you also solved structures of NSS bound to LC3, right?
Correct. That work was done by my collaborator, James Omnichisky, at University of Montreal. So he solved the crystal structure of NSS bound to LC3.
You can see the interface and how these amino acid, did you use that to determine which amino acids to remove?
Yeah, so initially we started on the computational side. Actually, we looked for the specific motif just by looking at the amino acid sequence. And then we used AlphaFold to predict whether it was in an intrinsically distorted region. That's really critical, that these motifs have to be in that very flexible region of the protein. And then when we looked at the crystal structure, you could clearly see that the one amino acid, that F261, was making key contact sites with LC3. And that provided evidence that we should go ahead and change that.
So did the crystal structure agree with the AlphaFold prediction?
It did. Yeah, but I would say the AlphaFold is not great at doing protein-protein interactions. At least the version I think we used 2 in that particular paper. It's getting better.
And when you do these experiments, I presume you're doing them in cells in culture, right?
Correct.
What kind of cells do you use?
We tend to use human small airway epithelial cells quite a bit because these are primary cells. They support the replication, and so a lot of times these types of cell processes, especially signaling pathways, they're normal, right? They're not, I don't prefer to use a transformed cell line, but we do occasionally confirm it in like a liver cells, like HUH7s.
So what would be a target cell of a virus in an animal? Would it be a respiratory cell or a liver cell?
It can impact the lung epithelial cells, but hepatocytes in the liver are the main target in the liver. In the brain, it's primarily the neurons.
And you could confirm these activities in other cells as well, right?
Absolutely.
Which is always good to do, right? I used to use HeLa cells all the time until people said it's irrelevant.
Why are you using those?
Because I got trained in a lab where they use them to grow a lot of virus, right? And but you can't do. I remember once I wrote an interferon experiment grant, and they said, "Why are you using HeLa cells?" And I never thought of it. He said, "Yeah, you're right." Okay. Does NSS do other things besides bind to LC3 and inhibit autophagy?
It does many things.
Yeah, that's why you like it?
I do. I think it's very multifaceted. We just had a review article accepted on this, and my student, who's the first author, Kaylee Petriccioni, she's also the first author of this other paper. She coined it the viral Houdini.
Viral Houdini.
Viral Houdini.
That's cool. Well, these are small virus genomes, and they don't have a lot of proteins. So one protein has to do many things.
It has to.
So can you tell me what else it does?
Sure. So another function that we found is that it will induce DNA damage signaling within cells. So for example, we have found that it will activate ATM signaling. So these are proteins that sense that the cell has its DNA damaged.
Right.
P53 signaling, which I think most people are very familiar with. So it activates that. But interestingly, we found that the DNA doesn't appear to be damaged. We couldn't detect damage. And so my hypothesis was perhaps it's actually sensing these filaments that NSS makes in the nucleus.
ATM?
Yeah, because it sits on promoters and these filamentous form. So that could be sensing that as damage, perhaps.
So normally, ATM would sense DNA damage that would activate P53, which would then put the cell into apoptosis, right? So that it's not duplicating damaged DNA,
exactly.
Right?
Yeah.
And so that's apoptosis, not autophagy. So is P53 activated, and is apoptosis also turned on?
You do see apoptosis, but
Notice
I know, I'm saying
it's okay. You can say whatever you want. It's nobody's going to agree.
It's about 2 days after infection is where we see a pretty big spike in cell death. And we hypothesized actually that NSS has a way of mediating this so that it kind of delays things, right? We don't get cell death right away, but it gives the virus enough time to at least go through some rounds of replication.
Okay. But it's interesting that it's doing it in the absence of actual DNA damage.
Mm-hmm. Yes, it is very interesting.
Have you looked to see if DNA is leaking out of the mitochondria? Because that could do it.
Yeah, that's a good question. I haven't, and I'm trying to think. I know my one colleague, Arthur Ryan at George Mason, was looking at some mitochondrial stress with Rift, and but I don't know if they looked at DNA leak.
So anyway, with dengue, that happens, and then that DNA activates interferon pathways,
For sure.
SING and C-gas and all that.
Yep.
But maybe it's doing a similar thing. I mean, activation of ATM in the absence of DNA damage is interesting. You're working on that, I guess, right?
We haven't followed up on that too much, I would
think it's
say.
cool.
Yeah.
Well, I happen to know it only because I teach transformation, which by DNA viruses, which involves p53. So that's pretty cool. What else does NSS do?
So it also, you're making me think back to my review article. It can also regulate a large number of signaling events within the cell. So we've done studies where if we use the virus that is lacking in NSS, a number of like kinases that all the signaling cascades, quite a number of those are no longer turned on. Cool. Where Rift really stimulates a lot of those. So
right now, are you still working on NSS in the lab? What kinds of questions are you interested in?
We're really interested in understanding if autophagy is regulating this filament formation. Or perhaps it's a dysregulation of autophagy that enables the filaments to form, because you shouldn't have these large filaments in your cells. Your cells should detect them, and you would expect them to be, shuttled to the autophagosome. And there's actually been studies that have shown that NSS is an amyloid-like protein.
Right.
Yeah. And so we really don't understand how these filaments exactly form, and why the cell isn't able to really relieve itself of them.
So if this happens in the brain, is that going to lead to dementias?
You would think. I don't think there's a lot of evidence so far that happens, but there's not, good studies to follow people long term, that have been infected with Rift and have developed encephalitic disease.
We just did a couple of papers on TWiV about herpes viruses and dementia. Big association there, right? Which is surprising to me, because I didn't know that there was such a brain involvement. Apparently there is.
Absolutely.
Because the vaccines really help you. So other viruses do go in the brain. It would be interesting to know. And amyloid is kind of an antiviral response, right, in the brain to make these proteins, but it has negative consequences.
Right.
Yeah.
Right.
So aside from RIF, what other viruses do you work on in the lab?
Yes, so we work on Venezuelan equine encephalitis virus and Eastern equine encephalitis virus. And, it's funny that you brought up this kind of long-term neurological damage, because that's a number of projects we have going on with those viruses.
Yeah, those are neurotropic, right?
They're very neurotropic, and it's documented that humans have long-term neurological sequelae with these viruses.
They also do with West Nile infection.
Right? Absolutely. We looked to a lot of the West Nile literature to understand how they've been studying it and the testing they've been performing.
Yeah, there are periodically outbreaks of V and E in the U.S.
Yes.
Right?
Yes.
Especially in Massachusetts, there's E, right?
Yes. I remember in 2019, they had a kind of a spike in the Eastern cases. I think there was maybe 26 that year, which was quite high.
And horses also will get it. And then when horses are involved, people get upset because they're expensive.
Absolutely. And people love their horses. So
here in Virginia, they have a lot of horses too.
Yeah.
I mean, XJ was telling me that the owners are very concerned about their horses.
They are. Yeah. In fact, a lot of non-scientists, they know about Eastern equine encephalitis virus because they're horse owners.
And West Nile too will also infect horses. So there's no veterinary vaccines for those viruses, right?
There are actually.
Yes.
And they're annual vaccines. But we still see that not everyone vaccinates their horses, and typically that's where you see the cases.
So I have one more question for you. As you know, that science is being attacked in the U.S., science and public health, NIH has been attacked. Universities are being attacked. And I wonder, where do you see this going? And, as a director of a unit that trains people, essentially, what do you tell young people about the future of science when it's being attacked and so many people just seem to not care about it?
It's a tough situation to deal with, for sure. I hope that I'm a positive role model for them in discussing science. We do encourage the students to communicate science. I know at least within the vet school, so I'm part of the vet school, they have to take a communicating science course to try to understand how they can communicate science. And different groups that I'm involved with here on the campus, they do have a large element of communicating science and as part of their kind of research program. I think that's one way that we can definitely do it. Also, it's just I think at this point, being very supportive to the students and the postdocs. Right. They're going through quite a bit right now. And I think we're all hopeful that things will change, but that doesn't mean that we don't speak up and we stay complacent.
I think also, if you meet with students and postdocs and discuss the situation, it helps a lot.
Yes. Yeah, they do. They just want to hear, what are your thoughts?
What do you think about it? What should I do, right? Give them advice. I think that's really important, not to just life as usual, do your research, but also you need to talk about it. But.
Yeah, and students are wondering, should I stay in science? Should I stay in viruses? And I try to, encourage them, of course you should stay. I mean, if you want to stay, you should stay. There will be jobs, you'll be able to figure it out.
You have to be persistent because it may take a while, right? You can't build something that's been destroyed overnight.
It's true.
Right? So you fire thousands of people, they're going to go somewhere else.
And they may not come back.
I mean, your lab, if you lose all your grants, then you let people go. Yeah. And even if you get them back in a month, those people are gone, right?
Exactly.
So I think that has to be discussed. But on a broader level, scientists have to talk to the public and say why it's good that we're doing this for you. Yeah. And that you shouldn't break it, right?
Yeah. Well, I will say that your keynote already inspired a number of people today.
That's good.
I had some people coming up afterwards saying that they really appreciated it and inspired the students and them to do better with communicating science.
Well, 2 students came up and said they wanted to do a podcast.
Yes, that's great. Yeah.
And they asked for advice. I said, well, you should get institutional support, from CEZAP.
Yep.
You could be an official CEZAP and you give them a place to do it, give them some funds to buy equipment.
Absolutely.
And say, we encourage you to do this so they don't have to do it on the sly.
Right? I agree. Yeah.
And
we will definitely support the students. Yeah.
I have heard PIs learn that the student or postdoc is doing a podcast and they're like, why are you doing this? You're taking away from...
Oh.
That's not right. It's important. It's just as important as doing the research is to talk about it.
Yeah, agreed.
So if CEZAP had a communication component, that would be great.
Yeah.
Okay. All right, that's TWiV from Virginia Tech. You can find this episode in the show notes at microbe.tv/TWiV. If you have any questions or comments, TWiV@microbe.tv. And as always, we'd love to have your financial support to do these programs. You can go to microbe.tv/contribute. My guests today, 2 guests in this program, XJ Meng and we get your Kylen Kenhal. Ken is in K-E-N. Thank you for sitting down with me. Yeah. And good luck with CEZAP.
Thank you, Vincent. It was a pleasure.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene Ramsey for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.